Welcome back, learners. So today in this video we will be discussing about your fermentation. Now, as you know that my main aim with this video is to describe your bioreactor design, but I will be discussing your fermentation. Okay. Now, because you have to relate this particular process with the bioreactor design. So we first need to understand the history of fermentation. So, once upon a time, many years ago, a man found a closed fruit jar. So a closed fruit jar was found by a man, and that particular closed fruit jar basically contained a honey bee. Okay. So when he drank the content, he tasted a new strange flavor. Suddenly, his head was spinning, he laughed for no reason, and he felt powerful. He drank all the liquid in the jar. The next day, he experienced an awful feeling. Okay. He had a headache, a pain, an unpleasant taste in his mouth, and dizziness. He had just discovered the hangover. You might be thinking that this is just a tale, it is just a story. But is it? Several archaeological excavations have discovered jars containing the remains of the wine that are 7,000 years old. So we have discovered 7,000 years old wine. Okay. So, and it is very likely that humankind's first encounter with alcoholic beverage was by chance. And how does this chance discover lead to the development of the beer and wine industry? And how did scientists eventually learn about the biological mechanism of alcohol production was a question. So for that, we need to move into the history of your beer and wine production, right? Now, you know that fermentation is a term which is associated with your beer, okay, which means to boil, okay. Now, when I'm talking to boil, that means there has to be some reason, some link that why we are saying that fermentation and what is boiling, these are two different terms, but how they are related. So, over the course of history, right, different trial errors and careful observation, different cultures begin producing fermented beverage. Mead or honey wine was produced in Asia during the Vedic period. So, during the Vedic period, that is about 1700 to 1100 BC, okay, and Greeks, Celts, Saxons and Vikings also produced this beverage. In Egypt, Babylon, Rome and China, people produced wine from grapes and beer from malted barley. In South America, people produce different beverages from grains or fruits, mainly maize. While in North America, people also produced wine, which is also known as your pulp, from agave, a type of cactus. So, different types of wine and alcoholic beverages were experimented upon by the people, by a common people. They are not researchers, they are not scientists, but they are common people. Okay. Now, you know that in the 17th century, so we will move on to the 17th century, okay, Dutch tradesman named Antony van Leeuwenhoek. I know you know the name Antony van Leeuwenhoek. They developed a high quality lens, okay, lenses, and was able to observe yeast for the first time. So yeast was observed for the first time. Okay. In his spare time, Leeuwenhoek used to use his lens to observe and record detailed drawing of everything he could include, very tiny objects like protozoa, bacteria, and yeast. Leeuwenhoek discovered that yeast consists of globules floating in the fluid, but he thought that they were mere starchy particles. So you know that they are globules like floating in the fluid, right? If you see like this in the microscope, what will you believe? You will believe it may be a starchy particle. And Leeuwenhoek was no different, okay? He also thought that it is a starchy particle, okay, of the grains from which the wort liquid obtained from the brewing of his whiskey and beer. Later, in 1755, yeast were defined in the Dictionary of English Language by Samuel Johnson. Samuel Johnson. Okay. So yeast was basically defined in the Dictionary of English Language by Samuel Johnson as the ferment put into drink to make it work and into bed to lighten and swell. So they say that this is a very good thing, that if you put it into a broth, it will make something which we can drink. If you put it into the bread, it will swell the bread, right? It will lighten and swell it. At the time, nobody believed that yeast were alive. They were seen as just organic chemical agents required for fermentation. Later, Lavoisier was also interested in analyzing the mechanism by which sugarcane is transformed into alcohol and carbon dioxide. So let's move on to another aspect, that is in 1789. So in 1789, French chemist Antoine Lavoisier was working on basic theoretical question about the transformation of substance. So transformation of substance was the question, okay? And in his quest, he decided to use sugar for his experiment, and he gained new knowledge about the structure and chemical reaction. He started using your sugar, and he started using the sugar and for his experiment, and also he derived the structure and chemical reaction. Lavoisier was also interested in analyzing the mechanism by which sugarcane is transformed into alcohol and carbon dioxide during fermentation. So he was also interested that how it is being converted to your alcohol plus carbon dioxide by your fermentation. Okay. So this lead to a development of your, fermentation and some development into the fermentation, right? Now, another important aspect came into being, that is in 1815, and that was one of the, very important aspect which we need to understand, and that is in 1815, there was a scientist. So in 1815, okay, French chemist Joseph Louis Gay-Lussac. Joseph Louis and Gay-Lussac. Okay, so these two have made some interesting observation about yeast. Gay-Lussac was experimenting with a method developed by Nicolas Appert, a confectioner and cooker, for preventing perishable food. Now, this is very important with the question boiling, right? Now, that they were thinking that how to prevent, that they are more important, and they're more giving more emphasis on your perishable food. And that this particular perishable food was actually rotting. That rotting is one of the mechanisms, that is your microbial food spoilage comes into play, right? So they were thinking that the method consists of boiling the wort in a vessel and then tightly closing the vessel containing the boiling fluid to avoid exposure of the air. So you take the wort, okay, the substrate, say, and you stopper it tightly, right? You boil it, right? You boil it and you keep it, okay? You store it. So what is happening when you are storing it? Because you have boiled this particular thing, the microorganism will be dying. And since the microorganism is basically dying, that is why, that you can store it for a longer period of time. So there comes the question of boiling, that if you boil wine, right, the wine will not be perished. That is what I am trying to say. And from there came the concept that farvier, that means to boil. If you boil it, it will enhance the shelf life. Okay. Next, you know that in 1835, Charles Cagnier de la Tour, a French inventor, observed that during alcoholic fermentation, yeast multiply by gamation. So, you know, you say budding yeast. So this particular budding yeast was also discovered. Okay. His observation confirmed that yeast are one-celled organisms and suggested that they were closely related to the fermentation process. And Theodor Kohn, Christian Arxebin, independently concluded that the globular or oval corpuscles which float so thickly in the yeast as to make it muddy while living organism. Burnett 1998. It was proved by Burnett 1998. Okay. Then came the very important scientist in the field of microbiology, and he has a huge contribution in various aspects of your microbiology. Okay. And that is your Pasteur. Okay. So French chemist Louis Pasteur was the first to demonstrate. So I will write down Louis Pasteur was the first to demonstrate experimentally that fermented beverage results from the action of the living yeast transforming glucose to ethanol. So glucose is converted to ethanol, okay, and that is basically being done by your Louis Pasteur, okay. And moreover, Pasteur demonstrated that only microorganisms are capable of converting sugar into alcohol from grapes, and the process occurs in the absence of oxygen. He concluded that fermentation is a vital process, and it defined it respiration. Respiration, please quote it, students. Respiration without air. Okay. So, Pasteur performed careful experiments and demonstrated the end product of alcoholic fermentation. Okay. Then, you know that in 1856, there is another significant discovery in fermentation. And the name is Bigot sought Pasteur's help. Bigot is another name, a man who sought Pasteur's help, and he was actually having a problem in his distillery. And what was the problem? The problem which produced, that is, he is trying to ferment, right, the sugar from the beetroot to alcohol. But he was finding something different. He was finding a sour milk. So why this was happening, he was unable to understand. Pasteur analyzed the chemical content of the sour substance and found that it contained a substantial amount of your lactic acid. Okay, so it was not ethanol at all. It was your lactic acid which was producing your sour milk. And when you get to see this organism in microscope, they're of course rod-shaped. They are not globular, right? And from there came another fermentation, another breakthrough, and that is your lactic acid fermentation. So, students, from the history, we come to know that we have two different major type of fermentation, that is your alcoholic fermentation and your lactic acid fermentation. So this is about the history of your fermentation and the concept that why it is called ferbeer, to boil, right? And I will try to acknowledge the source from where I actually made this lecture, and this is your By Nature's Education. This particular source is from a paper, Yeast Fermentation and the Making of Beer and Wine, which was published in Nature Education in Volume 3, Issue 9. Okay. So I will highly acknowledge that particular source from where I have got the information and have tried to deliberate you or percolate to the other individuals of the society. Thank you. Thank you once again. Stay at home. Stay safe.